All right, everybody. Hopefully the sound issue has been resolved. I would really hope so. I'm going to have lots of computers in front of me because we're going to have my phone as the broadcast for tonight. Well, welcome to Dr. D's Discussions, episode 16. I know this is the third time. Hopefully the third time's a charm. So episode is going to be Metabolism and Metabolomics. I know that you have to wave a lot now. But I'm sorry that we're not... It's tiny. I know it's not necessarily what we'd expected and what we wanted, but it's what we're going to have to deal with for tonight. Again, my apologies for the formatting issues and the late start. So just try to bear with us. We'll try to get through the slides a little quicker than we usually would. However, let's see what we can do. So metabolism. I know that I've read this before. Hopefully I don't have to read it again. But it's the sum total of all the chemical reactions involved in maintaining the living state of the cells and thus the organism. General metabolism is divided into basically two different groups. Catabolism, which is the breakdown of substances from a big molecule to a smaller molecule. And anabolism, which is the buildup of, smaller molecules to bigger molecules.
Sorry, mixed up in a body.
And, that kind of makes sense. If you've heard of anabolic steroids,
it's actually built from cholesterol molecules.
Yeah.
Principally, cholesterol background.
That's basically why it's steroids, basically. Yeah, that's why. And steroids are cholesterol molecules. So I hope the format's good. I have it on my computer. It sounds like it's going well. So, all right, metabolomics, what is that? It is the scientific study of the chemical processes involving metabolites. Specifically, it's the systematic study of the unique chemical fingerprints that specific cellular processes leave behind, the study of their small molecule metabolite profiles. Basically, all that means is that you're looking at every single chemical reaction that happens in a cell and thus the body. So it sounds like a lot of work. I know that you had already given your little opening statement, but I know I'm going to have to ask you to give it again. About how we look at little proteins and specific things.
So principally, as guys in molecular biology, we're looking at proteins and RNA and these types of molecules. But metabolomics is really I want to say like a finite mechanism of biochemistry where we're not so much involved with enzymes or proteins in a structure perspective, but we're looking at the product of these enzymes. So enzymes cleave and they're catalysts, they interact with other proteins, but they also take molecules and alter them. And those byproducts of those larger molecules are called metabolites. Mm-hmm. And that's what this is studying. That's what metabolomics looks at.
Yeah, and specifically I know that we're going to be talking about a lot of the things that happen inside of the cells, but a lot of metabolism happens to be, with xenobiotics. I know that I went through a little spiel in the first go around with this episode, but basically a xenobiotic is anything that's a foreign substance that you take into your body, whether that's food, drink, whether it's a lotion that you put on your skin, whether that's any type of thing that your body absorbs and does something with. Whether that's, as Lee said, breaking it down, building it up, and moving it from one thing to the next. So let's give a little bit of a breakdown of the two groups. Anabolism, making larger molecules from smaller molecules, so amino acids, nucleotides. Amino acids are making up proteins.
Right.
Nucleotides make up DNA, and then phospholipids make up the cell membrane. Smaller lipids make up, like cholesterols, make up steroids, different things like that is actually in the plasma membrane as well. And there's several different types of phospholipids and all these different
things.
Phosphatidylserine.
Yeah, they get crazy.
And then catabolism is basically larger molecules to smaller molecules. And the way I remembered it in undergrad was catabolism,
right.
Breaking it down. So that's basically the way I learned it.
So it's an interesting point to be made here as far as metabolism is concerned. You need fat in your diet. It's important. If you don't get it at all, your body will not be able to make cells.
No, at all.
We need lipids. And part of that is the metabolic process of taking fats that we get in our diet and reconstructing and reconfiguring those fats into usable things that our body needs.
So you're talking about those metabolites that we were
talking about those metabolites.
Those metabolites. Good old metabolites.
Good old MBs.
So yeah, I think that's a good segue into the next slide that we have. Nucleic acids, proteins, lipids. Oh my! I love that. You know, nice little what's it? Wizard of Oz reference right there for you guys. So displayed behind me is pretty much like the most simple, basic view of what happens in the cell in terms of building up proteins from amino acids to nitrogen pool, tissue protein, urea cycle. I know that we had discussed that in GC 550. And basically, if you have dysregulation of that, you get uric acid buildup. And in guys specifically, and actually in humans, not even like great apes, we're the only pretty much ones that actually have this problem, is gout and the buildup of like kidney stones and different things like that. So that's just something that you can have as a dysregulation of that system that can be looked at through metabolomics. Carbohydrates, the breakdown from glucose to
glucose or fructose or lactose or
acid,
any sugar molecule you can think of.
And then that gets broken down, or it gets built up to glycogen. So that's an example. Going to lactate is catabolism, and then making glycogen is anabolism.
Yeah, just to like be sure about what we're talking about here, these terms that lactose, for example, is glucose and galactose are two sugar molecules put together. To utilize that energy, though, you need to break them down into monomers, right? So that's catabolism, that's breakdown. When you have a lot of sugar, though, your body wants to do something with that, or it can store it. And one of the ways to store that is turning glucose into large chained molecules of sugar that we call glycogen, and that's stored in the muscle.
For the liver, yeah. And then another route that it could be stored in is if you have too much glycogen, it's putting it into reserves as fat.
Yeah.
So your body is a wonderful mixture of different things where it's got all these different pathways to, if you have too much of something, it'll turn into something else.
It's also a mixture of horrible things like reactive oxygen species, but we'll get to that later.
Yeah, but at the same time, all these three different macromolecular structures, or macromolecule groups, can interchange between the two. So you can go from proteins to making something for the citric acid cycle. You can do fatty acids to acetyl-CoA, which we know from beta-oxidative reduction, which is a fancy term for breaking fats down into something that can be used in the same pathway that glucose is broken down into for energy in the what's called the electron transport chain, which is
Krebs cycle all day.
Krebs cycle, baby, or citric acid cycle, whichever you like. And then from the citric acid cycle, those things get deposited into the electron transport chain in the mitochondria, which is your powerhouse of the cell. I know, I got hype. I got hype.
Dave had a 5-hour energy like 36 minutes ago, and I guess this is what happens when you do that.
Actually, I took a sip of one. Like, literally. Like a tiny sip. You're like, you don't need that? I'm like, no, I don't. So, well, basically, yeah, we can actually talk about that. That goes through the nucleic acid cycle because it's caffeine. Caffeine is a, is it purine?
Yeah, well, technically speaking, it's within those, it works on purogenic receptors, so it mimics
I know you did a
purines
nice little
do.
write-up on our
So I've got this article, Why Caffeine is My Best Friend. You should go read that because it's a love story everybody should read.
Yeah. That's a love story. It's like Leonardo DiCaprio and what's her face from Titanic? Philip Seymour Hoffman.
Yeah, well, I don't die in this one.
It's a Family Guy quote. All right. So I showed you this last slide where it was really quite simple in terms of the breakdown of proteins, glucose, and fatty acids. So right behind me I have, and I don't think you can really see it too well because of the lighting, but it's a whole goddamn thing, messwork of different nodes. So this right here, what I'm pointing at,
that looks like a circuit board.
Literally, it's insane how complex this actual issue is. And I'll throw up the slides in a little bit, but just to show you, this little area has about 60 nodes in it. And that's not even including all of the nodes that are necessarily there. It's rather fascinating to me that
these nodes represent actual
metabolites. So from one node, one little dot to the next dot is one metabolite breakdown. So there's an enzyme that's in between that stage. whether we know what that enzyme is, or we don't know what that enzyme is, or have it crystallized, it's something that's there. So if we know something's there,
Also, we don't.
yeah, we know something's there. It just may not be that we have it categorized, cataloged, and all those different fun, different things. How many isoforms everything has. So that's the other thing. Enzymes have different isoforms, so and different ways to break it and build it up and break it down, and you know, it's just mind-boggling as to the complexity of this field.
So the principal reason why we're even looking, or this field's even developed, is because we're trying to characterize disease in a bunch of different ways, right? So of course, genomics, which is the study of genetic variation within people, that was one way to look at things, right? What's going on at the genetic level that plays into disease, right? So then we started saying, well, let's look downstream of that. Let's look at proteomics. Oh, I lied. Let's look at the transcriptome, right?
Geez.
So let's look at the mRNA transcripts that come from the genetic material and see what's different there in disease, right? Then we start looking at proteins, proteomics. Now we're taking a step further. Let's look at the metabolomics. Let's look at the small molecular byproducts of all of these enzymatic reactions and see how this network correlates to the pathology of diseases. Can we use these things as biomarkers? Can we identify diseases?
Specifically the metabolites as biomarkers. So if you have an increase in, let's say, 1 through 100 of these metabolites, that can give you an indication as to the disease state of the individual, or it could give you an indication as to what you're predisposed towards.
Right.
And I just want to read this one thing from my notes, is that metabolomics, so named because it describes the metabolic products of the human genome, which is the genetics of pretty much everything that makes up a person, has existed at least as a modern scientific concept since Linus Pauling proposed in the 1950s that one could study breath condensates. So basically, you breathe out, and you could capture human physiology by that. So that's pretty fascinating that you can use somebody breathing out, catching what they breathe out, and determine what is going on, such as like a breathalyzer.
You know, people that have that too, and I don't know if anybody in the medical field will co-sign this. You know, people that have diabetes, they smell sweet, and because of the high sugar content in their blood, like they literally like smell like fruit. Like, yeah, it's an interesting thing. Yeah, because the sugar content is outrageous. Yeah, that's based on that concept and a whole bunch of other stuff.
I didn't know that.
Yeah, it's pretty crazy
cool.
actually. Yeah, so somebody co-signed that, nurses and doctors and everybody else watching this podcast.
Yeah, interesting. I know that you can smell bad and determine like somebody.
You know, you got halitosis and it's like,
ooh, yeah, you want to step back.
think something died in your neck. I don't know if that's actual disease or not. Probably.
Yeah, that's great. That's great.
Your tonsil is just rotting.
Yeah, I think you might have an infection.
Yeah.
Right. So.
So let's go to the next slide here.
Yeah, so I just put up another thing about how complex human metabolism really is and how it's all interrelated. And one big molecule that really sticks out is acetyl-CoA, and that's used
for everything.
Pretty much everything in the citric acid cycle. It's pretty much the starting point for the citric acid cycle. It goes back around, it goes back around, it goes back around. And it's pretty cool. Bioenergetics, speaking of acetyl-CoA, it's a term that describes the biochemical metabolic
There we go.
pathways by which the cell ultimately obtains energy. So that's brought within this field of study as well. What else do we want to say about metabolism? Anything in general?
I want to talk about a misconception that has to do with the whole alkaline diet thing, which actually is based off.
Got a good write-up on the website coming up. So
yeah, about that.
It's
actually based on a large misconception about this guy named Warburg, who there's actually.
I know we're going to be talking about that at the end. So do you want to say that for the cancer metabolism?
All right, I'll segue into the pseudoscience associated with this first, and then we'll get into the actual cancer metabolism about this. So my boy Warburg, back in like the 50s, started looking at associations between metabolism and cancer, and pretty much pioneered the field for cancer metabolism. And one of the things that gets taken out of context a lot is that cancer cells need an acidic environment to grow. And so if you drink alkaline stuff,
like baking soda.
yeah, or
and there was a a doctor. I know that I call myself Dr. D and I do these discussions, and I know I'm like the Dr. Phil of biomedical sciences here at all, but I'm not prescribing anybody anything. And I just want to be very clear that when Lee and I discuss something and we try to explain different things, we're not prescribing anybody anything. We're not telling you to go out and, you know, change your lifestyle for any reason. We're not trying to really. We want you to change your lifestyle in certain regards.
Diet and exercise, you can never say
we
that.
are not prescribing you to do anything. And I really want to be clear about that, because this individual was getting people who were terminally ill cancer patients to come to his ranch or his hideaway in Texas.
I don't know.
Whatever it was like the Playboy Mansion for pseudoscience, if you will.
Yeah.
But the completely worst thing in the world that you could actually do. He was getting these people to come and pay tens of thousands, hundreds of thousands of dollars to actually infuse them with saline, which is salt water, and baking soda. You know, baking soda is the thing that you put into your fridge so it doesn't stink. You know, bicarbonate, sodium bicarbonate.
Even though your body produces forms of bicarbonate in a way to neutralize chemical reactions, again, a lot of this stuff is based off, a lot of pseudoscience is based off tidbits of actual science information that get exploited to people that don't know the science, and the whole alkaline diet thing is an example of that. You can buy water today, it's got pH on it. That is pure marketing. They know it's popular, that's why they're putting it on there. And just to touch base on this thing with bottled water.
I like some smart water.
I do like smart water. I mean, it's got minerals in it, but
it tastes great, man. I don't know about you, but it tastes great.
But it's not helping my body with its pH balance.
No, it's not.
Yeah.
At all.
And so this gets taken out of context with regards to the whole acid thing. Cancer cells produce an acidic environment locally, and it's not a predetermining thing. That means that the environment doesn't become acidic, and that allows cancer cells to grow in it. Cancer cells, by way of their metabolic status, produce lactic acid, and thus the immediate area around the cancer cell is acidic.
But by infusing yourself with alkalinity is not going to do anything. Whether you put that into your blood, whether you eat something like an alkaline diet, there's things called a buffer, where it reduces the amount of change of pH. So your body has inherent systems, whether at the cellular level or the physiological, like clinical level, like such as blood, like Lee was saying, bicarbonate from the pancreas actually reduces the amount of acidity from when you actually have stomach acid and bile, and I think chum or chyme. It's actually chyme. Yeah, there it is. Chyme goes into the duodenum, which is the first part of your small intestine, and your pancreas spits out a bunch of bicarbonate to reduce the amount of acidity in that. Your body is never at equilibrium unless it's dead. And the thing about what we had said is, you know, the sum total of reactions in maintaining a living state of cells is they have these buffers so that you don't necessarily have a huge change of pH, so you die. If your body didn't have these buffers and you're trying to infuse yourself with this to like cure your cancer, it's not going to work.
You're going to die.
You're going to die.
Yeah.
So I know we're going to get into a little bit more of the Warburg effect at the end. So I want to get back to a little bit of this diverse array of applications that metabolomics can actually, be seen to do. It basically gives you the ability to extract value from other types of omics data like lipomics, and, genomics, and proteomics, and different things like that, to discover biomarkers, elucidate disease taxonomy, so the classification of diseases, and track treatment effects. So how actual real clinical trial treatments are actually going to affect the body in terms of if you give drug A, what does it break down into? And there's not just one step of breaking down, there are several states of breaking down. So you can go from drug A to like 100 different metabolites based upon that.
And in regards to this is pharmacokinetics, which is a very old field as far as pharmacology is concerned, but more importantly, pharmacokinetics is looking at the metabolism of a specific compound at a given time.
Yep.
Metabolomics is looking at the entire network of metabolites being produced by all of the compounds in the body at a given time for that patient, and then trying to associate that with the disease phenotype.
And that's not necessarily cause and effect, but correlative status. So it basically means that when you see A, B can be happening. And it's a good indication that when you do see A, that B is going to happen or could happen.
Could
it's
happen,
not
yeah.
necessarily that A is causing B, right? But it's just it could be the cause of, but however, it's just that when you study something, you're looking at the whole system as a whole, where you're not necessarily going to say when you pump a bunch of stuff into the cells or pump a bunch of stuff into the body, you see X outcome. That doesn't mean that these things necessarily cause X outcome, but that it could be an indication that those things happen.
So, I mean, big data is like a popular term, and I mean, no matter what field you're in, pretty much there's big data in every field, essentially. For science, for research, for biomedical science, it's big data in the form of these types of massive arrays where we're looking at huge amount of information in regards to metabolomics or genomics, whatever the case may be. I kind of feel that metabolomics is in its infancy as a study. And we should not necessarily be skeptical, but cautious of the conclusions that we are gaining from these types of arrays currently. Because even in genomics, which has been around longer, when you're screening for massive amounts of information, there's the possibility, and you're dependent on statistical analysis to provide conclusions, not necessarily validated results. There's a possibility of false positives and false negatives, and so when we're doing these screenings and finding out what role metabolomics plays, there's going to need to be a lot of validation in regards to these studies, which is really hard to do.
That is really true. So, I really do think that this is a great field in terms of the ability of information that's, or the amount of information that's going to give, that will actually give you, a little bit more context to the other fields that we just mentioned, like genomics and different things like that, proteomics. But I really don't think, because, and the reason why I'm skeptical of it as of right now, and the information that it's putting out now, is not necessarily that it's a faulty field in and of itself. It's just that we don't have the technology to look at every single different thing. So, like I had said, drug A can go to 100 different metabolites, and there could be several steps that are involved in that. And all these steps happen at the microsecond, nanosecond type of level, where they're transient, where you can have this thing happen, 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 and I just said that so everybody can hear and understand, but it's much quicker than that. It's literally, it's really difficult to observe, reproduce.
Yeah, I mean, you're looking at a snapshot anytime we do one of these assessments. It's actually one of the caveats associated with screening. Unlike genetics, which you're taking a snapshot, but that person's, genetics aren't going to change. The metabolomic profile of a person can change with their eating habits, exercise. Like, there are a multitude of things that affect the biology, which can then affect the metabolites that are being produced.
Actually, what you just said is something that Dr. Gertzman and Lewis, I just found this article from Harvard, the exercise effect, where you can have 10 minutes of working out, of exercise, and it changes 200 common metabolites. They're different levels, and an hour after this 10-minute little exercise, those levels are still not back to normal range. So, and then if you expand that, that's only 200 of the metabolites that you would see, common metabolites. If you looked at 2,000, 20,000, 30,000, what's the differences in ranges? And every single person is different. Every cell line is different. Having a jerked cell line in one lab and having them pass and pass and pass and pass are going to accumulate different mutations and have different parameters in terms of how they're going to function compared to maybe 1,000 miles away that's in a higher elevation or been handled a little differently or been given a different drug. Or,
all of these things are ultimately context dependent, and I mean that's the important thing here. I mean, as far as there has never been a decision where less information was better.
I like that.
Yeah, because if I would have known less here, I would have had the more information is better. So I am a fan of the way that this field is developing, and I'm a fan of the technologies that are results of being able to try to pursue this, because ultimately we're looking at a subset of biological processes here. But the collective approach is really where this is going to go. I think like computational chemistry was in similar aspect. Now most of our drug screening is done via like molecular modeling.
In silico, basically, what he means by that is that you have a compound A that gives a result. Let's say it helps cells, I don't know, fight off a disease better. You know, you can manipulate that compound A by changing just little molecules.
Right.
Like instead of having a CH group, which is a methyl group, here you change it here. You put it here, or you put an amine group there.
Let me predict those interactions using software. I mean, computational biology, which is now
it's a huge field.
yeah, it is a huge field. Pretty much, computational biology wants to predict biological effects or biological outcomes based on what we can predict from these biological processes.
I know the lab that you really want to do your thesis in, one I'm considering, does a lot of bioinformatic research. I
Right.
know, what is Dr. Santu?
Yeah.
His last name.
So, our homie, shout out to Santu, because I actually don't. We call you Santu, but your name is like 86 letters.
Oh my God. Guy's brilliant. He actually makes his own coding to actually come up with different ways to attack this problem in terms of an elevated protein this way
Right.
can be correlated with a depressed protein this way, or increased mRNA levels, decrease this,
Right.
increase that.
And he's actually mining data that's publicly
Available.
from hundreds of thousands of resources and screening techniques from all over the world that are compiled in these databases. But ultimately, what he does, guys like me come in the lab and they try to validate these conclusions that he's drawing using his data.
Exactly. So he does all the computer work and gives an idea, or maybe idea of maybe this explains what's going on.
Right.
And then Lee or I, as a hands-on biologist,
Or Bob.
or Arnold.
Yeah.
Dr. Claire Adams, brilliant woman. Love her.
Oh yeah, shout out to her too.
Yeah. I know that you're going to be doing a little piece on her. She's.
Oh yeah, we'll look out for that too. Women in science, we're definitely going to be pushing that too.
So I know that kind of gives us an idea of maybe what to look at to study with. So maybe that's what metabolomics can actually do. And I know that today's researchers recognize the relative contributions of genetics, behavior, and environment to maintaining health. But triggering metabolic diseases, on the other hand, remain unclear. What actually makes these things happen? So maybe by studying, metabolites through the course of maybe 20 years, 30 years of maybe one individual, two individuals, and then expanding that N out to
Right.
maybe thousands of individuals, doing like blood screens, breathalyzers.
It's all about building a complete picture here, and I feel like the same thing is happening. So epigenetics is also another booming field,
And actually, I have a little quote here about where is that? Epigenetics.
so I mean a lot of information is getting put out about epigenetics.
Metabolites can regulate epigenetic mechanisms and maintain the pluripotency of embryonic stem cells. It has been well established that metabolites such as ATP, acetyl-CoA, NAD, and S-adenosyl methionine, SAM, can function as co-substrates regulating post-translational modifications that affect protein activity.
Yeah, so I actually know this pathway specifically very well. SAM is a substrate used from. So when you eat folate, you produce SAM via
Folic acids.
Yeah, MTHFR, and SAM is used as a methyl donor for epigenetic modification.
Which is a reason why women who are pregnant take
folic acid. Yeah, well, specifically for spina bifida, which
has actually been decreased significantly because of
oh yeah, of course,
the infusion of folic acid or folates into our grain supply. So that's why you have cereals and breads with high folic acid content. Sorry for cutting off.
No, sorry, it's all great. The reason I brought up epigenetics is because there's a wide variety of information coming out, and we don't really understand everything that's going on there, and it's easy to draw conclusions off of a little bit of information when you don't understand the mechanisms.
Well, think about Dr. Mezo's lab, how they're coming up with a completely different theory or working.
Well, yeah, they have a hypothesis that would totally rewrite like the last 15 years of.
And I don't want to go into too much detail there because I don't want to get sued. I don't know what necessarily is confidential data. And
right.
That's not confidential data, but there's a lot of indications that he might rewrite the.
Yeah, so we'll plug it. We'll plug this cell paper that he has where he talks about it. So actually, you guys can read up on that. We'll let that let you investigate yourself.
But yeah, I really do like this, that, more information, like you said, is never bad. I really do enjoy that. I really like that quote. We should patent that. But let me give a little bit of a background about the Journal of Metabolomics. I know that it doesn't have that high an impact factor. I know it's not the sexy journal that Cell or Nature or Science. But
for our new followers, impact factor pretty much is how journals get graded. Whether it's their relevance.
Like how USDA grades meat, grade A, grade B, grade D, grade C. When you go to Taco Bell, it's edible. I would say metabolomics is like a C grade, so it's sold in chain restaurants.
Taco Bell, don't sue us. It's cool.
No, but no, it's a good journal. It has good science. They're pushing the envelope in terms of technology, and that's really what their main claim to fame is that they do push the envelope in terms of demanding more technology to dictate what they're actually saying. So I'm going to just give you the little bit of a snapshot of what I grabbed from their website. Metabolomics publishes a substantial share of the most significant current research in the areas of various technology platform development for metabolomics, metabolite target analysis, metabolic profiling, and metabolic fingerprinting. Progression in data preparation, storage, curation, and analyses, comparative integrated studies with transcriptomics and proteomics, including within a systems biology context, and the application of metabolomics as it relates to man, animals, and plants. And that's directly the description that Metabolomics gives about their journal. And basically all that means is that everything that we've stated so far about what it is and what they're studying is what they're trying to push and really try to bring to the forefront of science. Yeah. And I know that it's kind of in its infancy, just like genetics was and genomics was in the early 90s and
late
80s.
2000s and stuff,
But even the late 2000s, or early 2000s rather. But I really think that it's a field that's going to take off. And to be honest with you, especially given the fact that this,
my favorite,
cancer metabolism. So Lee, let's talk about what a tumor suppressor is and an oncogene is. I'll give the definition of a tumor suppressor, and then I'll let you.
I'll go ahead and just create some easy to digest examples of what these
I know that
things
we
are.
were taught the car analogy in class with tumor suppressors.
Yeah.
So a tumor suppressor is a protein, a gene that gives a protein, or something that could be an mRNA, microRNA, any type of substance that puts the brakes on the progression of cancer. So it suppresses cancer. So what it's designed to do is inhibit different pathways from going off the rails, or as I said, hit the brakes on a different pathway, like a car going down the highway, and to really slow it down and make sure it goes, okay, we need to slow down, the cell's not doing what it needs to do, we really need to check what's going on here.
Right.
Like an example, a big example that's usually mutated in a lot of proteins is, hey Evan, is P53 and retinoblastoma, RB.
Yeah.
So
P53 is the most well-known too. It's probably the most important because in half of all cancers, it's non-functional. It's mutated or it's not there anymore because cancers have figured out ways to delete this damn thing.
It's actually the reason why immortalized cell lines are immortalized.
Right.
That or I think it's ORF. It's either ORF or P53. That's the way that they keep.
ORF regulates MDM2, which regulates P53. They're usually ARF negative or P53 negative. But yeah, so oncogenes, so pretty much I like to use this example. If you're driving a car and you are at inherent risk for being in a car doing any miles per hour. Tumor suppressors are like seatbelts. You have a seatbelt on, it lowers your risk of dying. It doesn't necessarily stop you from injury, but it lowers your risk of injury. So tumor suppressors, pretty much, if you do get into an accident, if there is some type of insult to the body or to the car rather, then the seatbelt protects you.
Yeah, that's actually pretty damn good.
Yeah, and oncogenes, pretty much, they have normal functions. I'm going to throw out MYC, which is
your baby.
That's my baby right there. MYC is a protein that if you delete this protein, cells die. Embryos do not survive. People do not grow. Whatever.
They do not grow. They do not pass go. They do not collect $200. Many people have told me so.
Right. MYC is essential for life, right? So how can a protein that is essential for life also cause cancer? So when you overexpress, when you make too much MYC, it tells the cells to divide rapidly and uncontrolled. It's overexpressed in almost 40 to 50% of all cancers, and we can't do anything about it. Not yet, at least. Not until I discover a way to figure it out.
Yeah, I know you're gonna do big things with MYC.
But yeah, metabolomics plays into this, especially cancer metabolism, because one of the ways that you can modulate MYC activity is via modulating the ways that cancer cells utilize their energy.
Yeah, and I know that MYC's a big one. I know MAPK kinase pathway with KRAS and NRAS.
Oh yeah.
And all
I mean,
the RAS.
AKT, MEK, ERK. I mean, there's a bunch of these enzymatic pathways which
PI3K can be actually
Yeah.
constitutively active, and you know, basically what happens with cancer metabolism is that something gets rewired improperly so that it just goes,
right.
or there's no off switch. So basically, it's like having like you said, a car that just goes without ever being able to stop. And that means, and what we mean by stop and go is the proliferation and accumulation of more mutations, but the proliferation of these cells, which means these cells more rapidly divide than the neighboring cells. So that's why you get a tumor.
Right. Also, the reason why the cancer metabolism is very important here is because a cancer cell needs to create twice as many molecules as all rapidly dividing cells have higher energy metabolism than normal cells. They're dividing, right? And to divide, you have to make enough material that you can break into two cells and both be functional, right?
Yep.
So cancer cells have this ridiculous rate of metabolism because they're dividing at such a rapid rate. This alliteration. But yeah, so it's because of this energy demand. It's because of this energy demand that allows these cells to modulate these enzymatic pathways. So when we look at this from the therapy side, how can we utilize what we know about cancer metabolism because they have this unique energy demand to create drugs to stop them?
Yeah, and let's give a little bit more detail. I know that you basically covered everything I'm about to reread off my slide right here. How cancer cells reprogram their metabolism. Metabolic crosstalk allows for NADPH production and acetyl-CoA flux for lipid synthesis. So basically, you're increasing that mega molecule that we're talking about, acetyl-CoA. These metabolic pathways can be influenced by cell proliferation signaling pathways, which means, growth factors, different things. Activation of growth factor receptors leads downstream signaling cascade activation. Basically, you can have a growth factor receptor not have its ligand, what's called, and it just turned on. So basically, the protein is never off.
Yeah.
So it just keeps relaying signals like go, 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 go. It's like having your cell phone in the middle of class ringing uncontrollably. You can't turn the volume.
Yeah, no, and the off buttons don't work.
And you're like, you just throw it against the wall.
We use this term called constitutively active, which pretty much means enzymes have regulatory pathways that are part of their function.
As you can see behind us, if you can. I know it's a little bit blurry.
And those pathways, that regulation is dependent on the actual structure of that enzyme. So mutation can cause a change in that structure, and that allows the protein to function independently of its regulatory.
Yeah, and what I mean by a ligand is like how you stick a key into a lock and you turn it on or turn it off, like in your car. That's basically what we mean by turning something on. It just, like you said, changes the shape, and therefore changes the function. PI3K AKT pathway activation stimulates glucose uptake and flux to the early part of glycolysis. So basically, just making things go haywire. Another thing I personally really like to, what I'm going to really want to study is tyrosine kinase signaling. Negatively regulates flux through the late steps of glycolysis, making glycolytic intermediates available for macromolecular synthesis as well as supporting NADPH production. So basically all that means is you're having these growth factors, relay signals, and then these tyrosine kinases will increase the later portion of glucose breakdown and glucose synthesis to increase so that you can get more energy basically out of these molecules, or you can build up fats and you can build up different things like that.
And pretty much just to give an explanation to what tyrosine kinases are, tyrosine is an amino acid. They're part of proteins. This enzyme puts a molecule on these other proteins that either activates them or represses their function.
So a tyrosine kinase can be thought of as a relay race. So a 4 by 100 meter relay race team. So Lee, I, and 2 other people are tyrosine kinases, where I have the baton, I'm running, and I handed the baton off to Lee. Lee takes the baton, which is the protein, it's a phosphate group, so it gets stuck onto Lee. Lee goes, I'm going to run. He gives it to the next person. Then that person gives it to the next person and the next person.
Yeah,
so basically,
it's a good way to define a signaling cascade, which is how most of these pathways work.
And by having it constitutively active, is this relay race is never ending. So instead of a 4 by 100, we're doing like a 4 by 100 by 100 by 100 by 100 by 100 by 100.
16 squared, I don't know.
Yeah, I can't.
Yeah, ultimately the end product is cellular division, and these things just don't stop and result in cancer.
Yeah. So that's I think that covers a lot.
Yeah, that
was one heck of a wow.
Cancer metabolism is a huge field.
I know that we have a lot. I could go on for hours.
We could do like whole shows on cancer metabolism, actually.
Numerous shows on cancer metabolism. Basically, do we have anything else that we want to talk about in terms of metabolomics and metabolism there, Lee?
No, pretty much. We just really wanted to give an overview of what the field is and where it could go. As this year, so each year that metabolomics has pretty much become another extension of scientific research, we're seeing more and more publications associated with that field, and we'll definitely be ready to like update you guys with the latest and most accurate stuff associated with metabolomics.
Excellent. And I know that we did that pseudoscience article. I'm going to plug the heck out of that. Basically, we take down what we were talking about before with the alkaline diets and how yeah. But you got a little something?
Yeah.
Bless you.
Yeah.
Basically, and in the article, I give you 5 steps as to look out for fake news and fake science and how to really go, wow, this might be bullshit.
Yeah. Shout out to Ben Teller.
But basically that's our show for tonight. Next week we're going to talk about something nice, something simple. The eukaryotic cell, as simple as that may be. The eukaryotic cell is basically the cells that make up you and I, yeast, fungi, plants, protists like amoeba, and different things like that. And then malaria.
Malaria is a protist.
Yeah, it is a protist. Actually, it's not a bacterial word.
No, yeah, that's actually a common misconception. Malaria is transmitted by mosquitoes. It's not actually the mosquito; it's the small organism inside of the mosquito that gives you the disease.
And then we're going to do I believe in the first weekend of March is going to be what is cancer. I'm excited about that episode.
I'm super excited about that because there is just a plethora of misconceptions associated with what cancer actually is, and we'll be there to identify those things and make sure you guys are really informed so you don't get gimmicked into buying stuff that's not actually good for you.
Yeah, please. Yeah. All right, guys. So thank you for joining us for episode 16, Metabolism and Metabolomics. We'll see you next week. Bye-bye.
Later.